Welcome to the biology class, children. This is our third class, right? Children, you don't forget to write the lecture note and don't forget to draw the diagrams in our notebook. I am repeating the same in all my videos because it is very important. Most of the diagram based questions will come from this lesson. Okay, you should follow it. Okay, so let's start the class. Today's topic, pistil, megasporangium and embryo sac. Today we are going to discuss about pistil, megasporangium and embryo sac. Yes, we are going to discuss about the development of female gametophyte. We know that gynoecium represents the female reproductive organ and it consists of pistil. Pistil is bearing three parts: stigma, style and ovary. Stigma, it is a landing platform for pollen grain. It is followed by long and slender style and connects stigma to the basal part, that is ovary. So the basal swollen part, ovary, consists of a cavity inside it. That cavity is known as ovarian cavity or locule. Placenta is located inside this ovarian cavity. You remember the topic placentation? We discussed different types of placentation found among plants, isn't it? If any doubts are there, you refer our first class. Okay. And arising from this placenta, that is megasporangia, commonly called ovules. Okay. I will show you a diagram of placenta along with ovule. Arising from the placenta are microsporangia, commonly called ovules. The number of ovules in an ovary may be 1, like in wheat, mango, to many, like in papaya and watermelon. Okay, the ovule is attached to the placenta by means of a long stalk called funicle. So children, this is a vertical section through carpels. Here you can see a single ovule. This ovule is connected to the placenta. So here this is just a vertical section of ovule carpel. So here you can see placenta. So this ovule is attached to the placenta by means of a structure called funicle. F-U-N-I-C-L-E. Underline. Okay. Then the ovule body fuses with the funicle in the region of hilum. So this is hilum. So this hilum represents a junction between ovule and funicle. Clear? Ovule is well protected in a covering called integument. So here we can see integument. So in an ovule we can see both outer integument and inner integument. In future these integuments become seed coat. Okay. This integument covers the entire ovule except at the tip where we can see a small opening. It is called micropyle. It is called micropyle. M-I-C-R-O-P-Y-L-E. Micropyle. Underline. This micropylar end act as the entry point of pollen tube for the process of fertilization. Okay, so through this small tip, the pollen tube is entered into the embryo sac. That's why the covering integument is present all over the body of ovule except at a tip, and that area is known as micropylar region. While micropyle form one end of the ovule, chalaza is formed the basal end of ovule. Clear? So this is a structure of ovule. The same structure we can see in our textbook too. Okay. Then enclosed within the integument, a mass of cells called nucellus. N-U-C-E-L-L-U-S. Nucellus. We can see inside the ovule. It is made up of diploid, colorless, thin-walled parenchymatous cells containing abundant food materials. Children, in the nucellus of a matured ovule, there lies embryo sac, the female gametophyte. So children, this is nucellus, a mass of parenchymatous cells seen inside the ovule. One of the nucellar cell towards the micropylar end get differentiated into megaspore mother cell. See, this is the cells of nucellus, but one among them become megaspore mother cell, which can easily distinguished from other cells due to its large cells, dense cytoplasm and prominent nucleus. Isn't it? So how can we differentiate the single megaspore mother cells from other nucellular cells? It will be a large cell with dense cytoplasm and having a prominent nucleus. Children, during the development of male gametophyte, all the cells of sporogenous tissue are capable of become microspore tetrad. Do you remember that? But instead here, only one single cell become megaspore mother cell. Clear? The single megaspore mother cell undergo the reductional division meiosis for the development of megaspore tetrad. Okay, see, first megaspore dyad form, then megaspore tetrad form. Clear? Clear? So, out of the 4 megaspore tetrad, 1, 2, 3, 4 cells are there. Out of the 4 megaspore tetrad, one cell near the chelacell end, only the single cell near the chelacell end become functional megaspore. Okay, out of this 4 megaspore tetrad, only one at the chelacell end become functional and remaining 3 will degenerate. Okay, so now we are having only a single. I will show you another diagram. So listen, in this diagram, okay, in this diagram we can see a single functional megaspore. During the development there were 3, and in total there were 4 megaspores, but 3 degenerated. Okay, only one become functional. Clear? So now we are having only a single functional megaspore. So this functional megaspore undergo mitotic division, equational division. 3 mitotic divisions will be there. The functional megaspore divide mitotically to form 2 nuclei and they move to the opposite side of embryo sac. So this is the first nuclear division and form 2 nucleate embryo sac. Okay, it is followed by second mitotic division and form 4 nucleate embryo sac. We can see 4 nucleus here, 1, 2, 3, 4. First 2 nuclei migrate towards one pole, other 2 migrate towards another pole. And again, after the last mitotic division, 8 nucleate embryo sac developed. Clear? But it is very interesting to note that division of the nucleus does not trigger the cytoplasmic division and cell wall formation. So here we can't see any cell wall formation, only division has happened in the nuclear region. After this 8 nucleate stage, cell walls are laid down, leading to the organization of a typical female gametophyte or embryo sac. 6 of 8 nuclei are surrounded by cell wall. So here we can see total 8 nuclei, 1, 2, 3, 4, 5, 6, 7, 8. Isn't it? 6 of 8 nuclei are surrounded by cell wall. Here we can see a cell wall, here another cell wall, here also cell wall. And at the another end, here also we can see cell walls. Okay, so 6 of the 8 nuclei are surrounded by cell wall organized into cells. The remaining 2 nuclei called polar nuclei migrate into the center and they form central cell. Okay, now it is an organization of 7-celled 8 nucleate embryo sac. Now it become a typical embryo sac. Clear? So in the embryo sac, 3 cells are grouped together at the micropylar end constitute egg apparatus. So this region is known as egg apparatus where 2 synergids and 1 egg cell. Okay, these 2 are called synergid. I will show you one more diagram. This is the same diagram you can see in our textbook. This is the chelacell end. This is a micropylar end. At the micropylar end, we can see egg apparatus. Egg apparatus consists of 2 synergids and 1 egg. Okay, so in the synergids, we can see some cellular thickening. Can you see some cellular thickening here? Cellular thickenings. These cellular thickenings are known as filiform apparatus. Filiform. Filiform apparatus which play an important role in guiding the pollen tube into synergids. We know that the pollen tube releases two male gamete into the cytoplasm of the synergids. Out of two, one will fuse with this egg and it becomes zygote. And the remaining one fuse with the centrally located polar nuclei and in future it become endosperm. Is it clear? And at the chelacell end, there are three antipodals. They are nutritive in function, nourishes the embryo sac and helps in the growth and development of endosperm. So children, this is a diagrammatic representation of a typical female gametophyte or embryo sac. And children, please note that most of the question paper, we can see a repeatedly asked question. It is as follows. Draw a label diagram of a matured embryo sac. I noticed that most of the children lose mark in this area. You know the reason? The question is asked, most of them draw just an embryo sac. But if the question is asked to draw a typical embryo sac, you have to draw this diagram. That means the entire pistil you have to draw and you have to label, style, stigma and ovary. Inside the ovary you have to draw the embryo sac with three antipodals, egg apparatus and polar nuclei. Clear? And one more question was asked that draw an anatropous ovule. You know what is anatropous? Anatropous means inverted ovule. So you have to draw a diagram with the micropyle is bent down to the funicle to which the body of the ovule is united. Clear? Okay children, today we discussed the development of female gametophyte, embryo sac. One among the nucellar cell become functional megaspore and they undergo meiosis and form megaspore tetrad. Out of the 4 megaspore tetrad, 3 will degenerate and 1 become functional megaspore. They undergo 3 continuous nuclear division mitosis and form a 8 nucleate embryo sac, and they rearrange 3 at the chelacell end, 3 at the micropylar end and 2 at the center. That is a typical female gametophyte embryo sac. Okay. So if any doubts are there, you just ask me and read the textbook thoroughly, draw each and every diagram and answer the questions. Okay. We can see in the next session. Till then, bye.